Heterosis in plant breeding. In plant breeding, heterosis, or hybrid vigor, refers to the phenomenon where hybrid offspring exhibit superior performance compared to their parents. This improvement can manifest in various traits, such as yield, resistance to diseases, or stress tolerance. Heterosis is widely utilized in plant breeding programs to develop high-yielding and resilient crop varieties by crossing genetically diverse parent plants. Mid-parent heterosis, hybrid performance compared to the average of both parents, heterosis. High parent heterosis, performance compared to the better performing parent, heterobeltosis. Economic heterosis, performance compared to the commercial variety, commercial heterosis. Mid-parent heterosis, the heterosis percentage can be calculated using the formula, the difference between the F1 hybrid value and the mid-parent value, divided by the mid-parent value, and then multiplied by 100. Better parent heterosis, the difference between the F1 hybrid value and the better parent value, divided by the better parent value, and then multiplied by 100. Commercial heterosis, the difference between the F1 hybrid value and the commercial variety, divided by the commercial variety value, and then multiplied by 100. Commercial heterosis, the difference between the F1 hybrid value and the commercial variety, divided by the commercial variety value, and then multiplied by 100. This is an example: when the hybrid value is 40 and the commercial variety value is 35, the commercial heterosis will be 14.28%. The genetic basis of heterosis, or hybrid vigor, is explained by several key hypotheses, each providing insight into why hybrids often outperform their parental lines. The dominance hypothesis suggests that hybrid vigor arises when dominant alleles mask deleterious recessive alleles from each parent. This masking effect reduces the likelihood of harmful recessive traits expressing themselves, resulting in a hybrid vigor. The overdominance hypothesis, on the other hand, posits that heterozygosity at certain loci provides a performance advantage. According to this hypothesis, specific combinations of alleles at a gene locus work together to enhance traits beyond what either parent alone could achieve. Finally, the epistasis or interaction hypothesis suggests that hybrid vigor results from complex interactions among multiple genes, where the combined effects of genes across the genome contribute to the hybrid's enhanced performance. These gene interactions can lead to synergistic effects that produce unique and favorable traits in hybrids, which neither parent individually expresses. Breeding strategies to exploit heterosis. Hybrid breeding, creating hybrids by crossing inbred lines to capture heterosis. Marker-assisted selection, MAS, using genetic markers to select parent lines with desired traits. Genomic selection, GS, predicting hybrid performance through genomic data. Applications of heterosis in crop improvement. Heterosis exploited for yield improvement. Examples of heterosis exploited for yield improvement: maize, rice, and wheat hybrids. Hybrid vigor used in horticulture for uniformity and vigor. Adaptation to environmental stress, hybrids for drought tolerance, disease resistance. One major obstacle is the high cost of hybrid seed production, as creating and maintaining inbred lines for consistent hybrid performance requires significant time, labor, and resources, making hybrid seeds expensive for farmers. Additionally, genetic and environmental variability poses a challenge, since hybrids may perform well under controlled conditions but exhibit inconsistent results in diverse field environments. This variability affects the reliability and adoption of hybrid crops across different regions. Lastly, despite advancements in genetic research, the complexity of heterosis genetic basis remains a challenge in some crops. Fully understanding the underlying mechanisms of heterosis, such as the roles of dominance, overdominance, and epistasis, is essential to predict and optimize hybrid performance accurately. This lack of understanding can limit the ability to harness heterosis effectively, especially in crops with complex genomes, ultimately impacting the development of consistently high-performing hybrids. Future prospects. Advances in genomics and biotechnology. Genomic prediction models. Epigenetic studies. In conclusion, heterosis plays a vital role in plant breeding, offering significant benefits such as improved yield, resilience, and adaptability, which are essential for enhancing crop performance. Continued research into the genetic basis of heterosis is crucial to fully harness its potential, enabling breeders to develop sustainable crop varieties that support global food security. Advancing our understanding of heterosis will ensure that agricultural systems can meet the demands of a growing population under changing environmental conditions.